Hello students, welcome back. Welcome back to my biology online classes. In my previous lectures, I have shown that how RTK is being activated. RTK full form Receptor Tyrosine Kinase, and after being activation, what happens? There are several types of proteins with which the activated receptor may interact. Some of these proteins are GAP. GTP is activating protein. PLC gamma phospholipase C gamma. SRC like non-receptor protein tyrosine kinase. The activated RTK can interact with these proteins GAP, PLC gamma and SRC like non-receptor protein tyrosine. Activated RTK can induce signaling pathways. Ras-Map kinase pathway, IP3 DAG pathway, PI3 kinase. Signaling pathway can be initiated after the activation of receptor tyrosine kinase. Now let's concentrate about this pathway. The first one is Ras-Map kinase pathway. Signal transduction pathway. What is Ras? It is an intracellular. This is present inside the cell. Monomeric. As all of we know what is monomer and what is dimer, how it is formed in case of RTK, I have already discussed with you. Monomeric GTPase switch protein. GTPase, as this term is present, means surely this is an enzyme, and GTP means this enzyme will go to break here, dissociate GTP. Okay, GTP, as the name suggests, triphosphate. There are presence of 3 phosphates, and after this ACE activity, surely it gives rise to GDP. Okay, it's a common conception. Guanine triphosphate, 3 phosphates are there. After this ACE activity, GTP, after this GTP ACE activity, what will be occur in case of GTP? GTP will dissociates into GDP plus an inorganic phosphate, and for this process, all we have to know that energy will released, and that energy is required for this signaling pathway. Functions as transducing signal. What is this transducing means? Transducing means it increases the length many more times. If 10 molecules are coming, after transducing, these 10 molecules will be 10 to the power 10 from RT. This Ras, as it is GTPase switch protein, and it switches between GDP to GTP form. So Ras cycles between inactive GDP bound form to active GTP bound form. And this Ras cycling, this requires 2 proteins, GEF, GAP. The full form of GAP is GTPase activating protein, and full form of GF is guanine nucleotide exchange factor. So for this cycling, 2 proteins are required, GAP and GAP. Full form of GAP is GTPase activating protein, and full form of GAP is guanine nucleotide exchange factor, which bind to Ras GDP complex and causing dissociation from the RAS from this GDP, because GTP present in higher amount than GDP in normal eukaryotic cell. GTP binds promptly empty RAS molecules. Now RAS molecules is empty and the lots and lots GTP, they are going to rush through this empty RAS and bind it, and this occurs with the release of GEF protein. Normally the average lifetime of GTP bind to RAS is about 1 minute, very fast reaction. GAP, now come to GAP, binds to RAS GTP and this accelerates its intrinsic activity. Binding of a ligand to an RTK leads to activation of RAS. Two cytosolic protein, adapter protein and GE provides the link between RTK and RAS. Now let's draw the diagram. Make this plasma membrane layer as like this fashion, okay? PLP layer. PLP layer of plasma membrane. First of all, they are in monomer form. GDP is bound with RAS, inactive. Ligands are coming. Ligands are coming and going to attach here. Obviously, this is cytosolic. This one is cytosolic domain, and all of these are monomer form. Both of two are monomer form, is exterior. Now the next step, dimerization. Is bind here and it is phosphorylated. It is phosphorylated, and here dimer formation. This one is now dimeric receptor, okay? GDP RAS, GDP RAS, till now it is inactive, okay? After that what happens? Binding of GRB2 and SOS couples receptor to inactive RAS. These are the three steps. What will go to binding? Binding of GRB2 and SOS. Now the question arises, what is GRB2 and what is SOS, which are going to couples receptor, okay? So what is GRB2? GRB2, here adapter protein is required in this stage. What are adapter? Here normal function of adapter. Adapter means in case of our mobile charging, whatever charging, we know that it is a link up between our device and electric source, okay? It acts as a link and adapter. So here protein, adapter protein, here adapter protein also act as a link of power supply. Here not power supply, and some it helps some supply. Here it helps for some connection. They are known as adapter protein. Okay? And these adapter proteins bind to an activated receptor. And after binding of this adapter protein, some other proteins also go to bind here. And after this binding, many become substrate for this receptor. But these adapter proteins, adapters has no catalytic power, but they serves to bring other proteins to the receptor. Okay, it has no its own catalytic power. What will be its function? It serves to bring other proteins to the receptor. Here the cytosolic adapter protein is GRB2. So now let's clear what's the function of GRB2. It acts as an adapter protein, okay? binds to this GDP. We'll go to bind to this GDP. So it binds to a specific phosphotyrosine on an activated ligand bound receptor and to the cytosolic GEF SOS protein. So this one is cytosolic GEF protein SOS, and SOS then promote the formation of active RAS GTP. From this inactive RAS GTP to active RAS GTP. The full form of GRB2 is growth factor receptor bound protein 2, and full form of SOS is zone of 7 less. Full form of this GRB2 is growth factor receptor bound protein 2. I am not going to make it a separate set. Yeah, third picture I am going to describe here. GDP RAS inactive form, SOS and GRB2 bind to this. This GDP is now changed to GTP and it being active RAS right now, and start to give signal. And make sure draw this in, this one is first set, second set, and make this in third set. First make this GDP inactive form, and in the fourth step make this GTP active and give signal. So this is Ras-Map kinase pathway, and SRC homology domain, yeah, SH2 domain, SRC homology domain, yeah, SH2 domain in GRB2 binds to a specific phosphotyrosine residue in the activated receptor. And then what happens? The SH2 domain, this was originally identified as a conserved amino acid sequence. Conserved amino acid sequence domain means its amino acid sequence is conserved in this domain. Any activity occur in this domain due to this conserved amino acid sequence, very specific conserved amino acid sequence in the non-catalytic region of tyrosine kinase. Tyrosine kinases encoded by viral oncogenes. And what is this viral oncogenes? It is a separate, totally separate concept, and I am not going to discuss it here. Such as VSRC, SH3 domain of GRB2 binds to SOS. This SH3 binds to protein rich motif. So this is all in a nutshell, very important Ras-Map kinase path.